Good morning, everyone, and welcome to today's AAPS eChalk Talk. Today's eChalk Talk is hosted by Sapient. You may submit your questions at any time by entering them into the Q&A box of the Zoom platform. All questions will be answered at the end of the program. I'm happy to welcome you to today's talk entitled Differential Disease-Modifying Tractable, Identifying Optimal Drug Targets with Discovery Proteomics and AI. The number of tractable targets is rapidly expanding thanks to new drug modalities opening the druggable space from a few hundred to several thousand proteins. The challenge now is comprehensively characterizing the expanded breadth of potential protein targets to identify the best target for a given therapy. This webinar will discuss how mass spectrometry-based discovery proteomics combined with AI-based target validation enables direct, deep interrogation of the proteome to identify differentially expressed disease-modifying tractable protein targets to match with a given drug. And now I'd like to introduce our speaker, Dr. Mo Jain, founder of Sapient. Dr. Jain is a physician scientist with more than 20 years of expertise in physiology, computational biology, and mass spectrometry-based bioanalytics. He formed the Jain Laboratory at UCSD, where he led his team to develop next-generation mass spectrometry systems to probe the non-genetic landscape of disease across large-scale human studies. Dr. Jain founded Sapient in 2021 as a spin-out of the Jain Laboratory to expand upon this mission, providing bespoke services, multi-omics, biomarker discovery. And with that, I'd like to turn it over to Mo to start our session. Mo, the floor is yours.
Thank you so much, Stacy, and thank you to the AAPS. We're very excited to be here today and really welcoming all the audience and really excited to talk through how we leverage mass spectrometry-based proteomics together with AI now for really next-generation target identification and validation. So as the audience is aware, traditional drug targeting has really focused upon a very small panel of targets as a whole. And this is really owing to the fact that the vast majority of drugs developed today have really involved small molecules. Even since 2017, with the introduction of biologics and other modalities, still more than 50% of drugs that have been developed really are leveraging small molecule biology and chemistry. Because of this, the actual tractable space, the amount of the druggable the amount of the genome or the proteome that is druggable has always been quite limited. And if we look across all FDA-approved drug targets today, both human and bacterial, there's less than 1,000 proteins that have been targeted by particular therapeutics agents. Now, there's been a massive transformation in the modalities under which we can drug various targets over the last half decade. This includes radioligand pharmaceuticals, targeted protein degraders, cell engagers, gene and cell therapies, CRISPR-based reagents, ASO, and other knockdown reagents, as well as ADCs. Now, collectively, the result of this has been that the druggable genome has gone from 900 or so proteins to now over 20,000 proteins, over 50,000 proteoforms. And so the discovery opportunity has massively changed and expanded at a rate that has been unprecedented. Now the challenge has evolved. It is no longer whether or not we can target a particular protein, but rather what is the best protein to target, and how do we validate those particular targets? Now, to date, when we think about traditional target identification, this has largely occurred through surrogate measures. As mentioned in the prior slide, the vast majority of therapeutics are targeting proteins, and this is where the business end of drug interaction occurs. The way we've gone about identifying these targets, these proteins, for therapeutic intervention has largely come from DNA and RNA-based measures using genomics and transcriptomics-based technologies as surrogate markers for those proteins that are altered in particular disease states and hence targets. The challenge is that proteins relative to their RNA are very poorly correlated. If we look at a healthy or homeostatic or normal state, there is for many proteins a rough relationship with the amount of mRNA for that particular protein. However, if we look in a diseased or perturbed state after therapeutic intervention, the relationship between the amount of RNA and protein levels for any individual target is quite dysregulated. And this poor correlation means that using DNA or RNA as a surrogate marker to identify the ideal protein targets becomes quite problematic. Now, traditionally, we have used mass spectrometry as a means for identifying and measuring proteins. Now, there's particular challenges and there's advantages to using mass spectrometry. When we think about the advantages of mass spectrometry, they allow for direct peptide sequencing, so we can measure proteins with very high analytical specificity. Not only can we measure the host or native protein, but we can also measure the variants in that protein, the isoforms, the truncated proteoforms, etc., as well as the post-translational modifications, which in many ways represent the business end of protein biology. Proteomics by mass spectrometry is a non-targeted or can be done in a discovery means, whereby we're not targeting certain proteins, but rather we can measure all the proteins that are altered under a particular disease state. Mass spectrometry is particularly applicable to multiple sample matrices, whether it be human samples or preclinical samples or cellular samples, whether they be solid samples such as a tumor, a tissue, a cell, or an organoid, or it would be a liquid sample such as plasma, serum, urine, CSF, tears, eye fluids, or media. Mass spectrometry enables the analysis of all of those matrices. In addition, mass spectrometry can be highly operationalized in regulated environments, including CAP and CLIA, which enables rapid clinical translation of discoveries and markers that are discovered through mass spectrometry. Now, while these advantages have always placed mass spectrometry as the forefront or the go-to gold standard for protein measurements, there have been very important limitations. First and foremost above these, traditional mass spectrometry has not been robust enough or scalable enough to really enable meaningful discovery, particularly for identifying of new drug targets and validating these targets. Fortunately, that has all changed over the last 12 months or so, in which there's been a number of innovations, both in sample handling and mass spectrometry hardware and mass spectrometry software, all of which collectively now make mass spectrometry really a de facto means for measuring proteins for target identification. These innovations have greatly expanded scalability, allowing for the robust interrogation of thousands of biological samples at a given time, at a depth that's required, measuring thousands of proteins, proteoforms, and PTMs in each of these thousands of biological samples. And this is what's really required in order to identify and validate targets. In addition, mass spectrometry has expanded greatly with label-free quantitation, which allows for very active and deliberate discrimination of protein amounts, both in healthy tissues as well as diseased tissues, which is also fundamental in order to identify the ideal targets. This is where Sapient really was born, as a means for providing services in high-throughput discovery mass spectrometry. Shown here is a video of our laboratory, or a portion of our lab. We're based in San Diego, California, and you very quickly get a sense for the amount of state-of-the-art mass spectrometry technology, hardware, automation, and innovation that's at the heart of Sapient. Much of our proteomics technologies and tools leverage state-of-the-art instruments, including the Bruker timsTOF HT and Pro2 lines of mass spectrometers, which are really high-resolution ion mobility QTOF instruments that are absolute ideal for these proteomics measurements. Our systems allow us to take any biological specimen, whether it come from a human, a preclinical model, or a cellular system, again, whether it be liquid samples such as plasma or urine, or a solid sample such as tumor or tissues or cells. And in that singular sample, we can assay thousands of proteins, metabolites, and lipids across virtually any matrix and any sample type. We operate under GCLP, but given our targeted approaches here and regulated environment, we operate as well under CLIA and CAP, and this allows us to rapidly translate any of our discoveries with rigorous QC. Now, within the proteomics space, there's really three main sources of innovation that have enabled us to finally be able to do robust target identification and validation using these workflows. These can be summarized as sample handling and extraction, mass spectrometry hardware, and mass spectrometry software. Again, collectively, these systems are now enabling the analysis of thousands of samples with measurements of tens of thousands of proteins and proteoforms in each of these biological specimens. When we think about sample handling and extraction, we've developed nanoparticle systems here that enable us to isolate not only free-floating proteins, but also membrane-bound proteins, including those in extracellular vesicles and in the exosomes, particularly from a liquid sample such as blood or urine. This allows for de-enrichment or depletion of high abundant proteins such as albumin that can obfuscate the measurement of lower abundant proteins while simultaneously enriching lower abundant proteins. From a hardware perspective, we're leveraging the Bruker-Timstorf HT mass spectrometer and coupling this with EvoSep nanoflow chromatography. This enables the most rapid and robust label-free DIA discovery proteomics. The label-free nature of our proteomics really allows for discreteness with quantitation and the most accurate measure and precise measure of these proteins. From a discovery proteomics perspective, these proteins are isolated using our nanoparticles, digested into their peptides, and then each of these peptides is sequenced. This direct peptide sequencing together with state-of-the-art software now allows for very, very high confidence in protein identification, typically much higher than is what observed with aptamer or antibody-based proteomics approaches, as well as enable the measure of proteoforms or isoforms of proteins and post-translational modifications. When we think about the post-translational modifications that are assayed by these systems, they include phosphorylation, acetylation, methylation, ubiquitination, as well as in cells and tissues, sumylation, glycation, and other critical post-translational modifications. As mentioned earlier, these workflows can be applied to virtually any sample, matrix, and modality, from a human specimen to a preclinical specimen to a cellular model, for both liquid as well as solid samples. Importantly, particularly with regards to plasma proteomics, because we're assaying using our nanoparticle systems, not only free-floating, but also exosomal, EV, and membrane-bound proteins, we're able to capture many of the biologically important proteins, including over 1,700 membrane-bound proteins. This becomes particularly critical as over 70% of emerging therapeutics target membrane-bound proteins. And so being able to measure these in a liquid sample such as blood becomes essential for target identification and validation. Now, when we generate this data, it's critical that we be able to isolate this and from this identify the optimal drug targets. And this is where the AI platform that's central to Sapient becomes absolutely critical. When we think about how we classify optimal drug targets, it really falls into 5 categories that are listed here. The first, we're looking for those proteins that are differentially expressed, that are either uniquely expressed or overexpressed in a disease tissue compared to its normal counterpart. Secondly, we look for those proteins that are disease modifying, that are mechanistically involved, both in the biological and signaling pathways that are central to the disease process. We look for what's called therapeutic tractability. Can we assess this particular protein by a given therapeutic drug in the specific cell type or tissue of interest? We look for what's called off-tissue effects. We heard about off-target effects. With emerging therapeutics, we think a lot about off-tissue effects, meaning on target, but in a normal tissue that's different than the therapeutic targeted tissue. This becomes very important in thinking about adverse potential events that may be associated with these emerging therapeutics. And then finally, we think about those particular targets and markers that enable patient stratification that can be measured, particularly in plasma in humans, as companion diagnostics that enable optimization of clinical trial enrollment or clinical trial implementation. So to walk you through some of these and to double click into this area, when we think about differentially expressed proteins, we're really looking for those protein and proteoforms that are uniquely or highly overexpressed in a particular disease state. Shown on this volcano plot on the left here is a representative example of a tumor relative to its corresponding normal tissue. And what we can find among the thousands upon thousands of proteins that are assayed, over 12,000 in this individual experiment, we find that a number of them, over 30% of proteins, are differentially expressed or altered at a false discovery rate below 0.05 in a tumor specimen relative to its normal counterpart. Now, among these differentially expressed proteins, we can begin to, again, understand those signaling pathways and processes, the subcellular localization, and the cross-validation with orthogonal disease states that are central to identifying disease-modifying aspects of these proteins. Shown here is an example of an enrichment for different signaling pathways and biological processes that are specifically altered in this tumor specimen, again, relative to its normal counterpart. And we can find very well-known signaling pathways, such as apoptosis, mitotic cell signaling, and mTOR signaling. We also find other less well-appreciated signaling cascades, which are also central to this process, including the unfolded protein response, UV response, interferon alpha response, and estrogen response. This becomes very important in understanding the underlying biological mechanisms that may be deriving a disease state or driving a disease state, and also identifying these protein targets that will be modifying for that particular disease. In addition, we can think about drug targets. So overlaid on this volcano plot are the hundreds of known drug targets, each shown up as an orange dot. You can find very well-known drug targets that are labeled on this image here. And interestingly enough, you can find that many of these drug targets are highly differentially expressed, either overexpressed or underexpressed in this disease, in this tumor of interest. Now, interestingly, if you look and see among these most differentially expressed proteins, we also find other proteins that have not traditionally been targeted that are similarly expressed or differentially expressed to the same degree as known drug targets. This becomes really, really important and for us to be able to identify what may be an optimal drug target in comparing it to existing drug targets. When we think about therapeutic tractability, we use AI-based classification to really understand not only the differential expression of a protein relative to normal counterpart as well as other normal tissues, but also to understand the potential therapeutic intervention, meaning can this protein target actually be accessed by a particular therapeutic intervention? Central among this is really being able to identify those proteins that lie at the cell surface layer. These have become essential or central to radioligand pharmaceuticals, ADCs, T-cell engagers, gene and cell therapies, as well as other emerging therapeutics that really depend upon accessing these particular targets at the cell surface layer. Now, we've developed here at Sapient an AI classifier that uses a number of biological information in order to be able to assess whether a particular protein is highly and differentially expressed specifically at the cell surface layer. This includes chemical tagging data, antibody-based protein imaging, protein topology, organellar localization, gene ontology, protein sequence, and even protein phylogeny that collectively are fed into our underlying AI-based classifier. And from this, then, we can provide a statistical value for what the chance is for a particular protein of interest to be highly expressed and differentially expressed at the particular cell surface layer. Now, in addition to assaying and understanding these particular targets in the tissue of interest, it's also absolutely essential that we understand off-tissue effects. As we've seen in a number of these emerging therapeutics, they can be extremely potent in targeting an oncology cell or a tumor cell or an autoimmune cell, et cetera, in a particular tissue. But many of these therapeutic agents also have very important on-target off-tissue effects, meaning they're also interacting with that same protein in an orthogonal normal tissue. So as part of our AI classification around therapeutic tractability, we've also developed very large databases around protein levels in normal human tissues. Shown here on the slide is over 20 normal human tissues in which we have dense proteomic measurements. And so for any therapeutic target, we can begin to evaluate their expression in these normal tissues. From this information, we can begin to understand potential adverse or off-tissue effects that may occur from any therapeutic, particularly in the oncology space. Now, finally, when we think about protein measures, it's really critical that we think about this translational gap. How can we leverage this information and this particular protein measurement to optimize not only for the development of the target, meaning development of the therapeutic that will be targeting that particular protein, but also subsequently how we use these protein measurements for optimization of clinical studies? Now, when we think about typical clinical studies, a group of individuals with a particular disease diagnosis are all given an intervention. And as we're all aware, drug development is fraught with failure at a clinical stage. When this occurs, particularly at a terminal phase 3 stage, it can be extremely expensive, both in time as well as finances. And it's essential that we be able to leverage this protein measurement to optimize both for clinical trial enrollment, meaning identifying those patients who are most likely to benefit for a therapeutic, as well as for monitoring of a particular clinical trial for companion diagnostics. You can imagine that if there's a good protein target here that can be assayed from plasma, we can begin to evaluate target engagement, pharmacodynamic profiles, and companion diagnostics that allow us to stratify individuals into those that may benefit from a particular therapeutic versus those that may not benefit as much from a particular therapeutic. This becomes essential for enrolling ideal candidates into a clinical study and limiting the expense and time required for clinical studies. As part of these efforts to identify and optimize validation of particular targets, Sapient has developed its own internal data assets, including our tumor database that now spans more than 1,000 tumors, in which we have matched genomic sequencing, RNA sequencing, and mass spectrometry-based proteomics, as well as clinical outcomes. In addition, we've developed very critical normal human tissue data sets that span over 20 plus different tissue beds in over 1,000 individuals. The tumors that we oftentimes think about are those common ones that are listed here in the middle column, including laryngeal carcinoma, pancreatic cancer, serous carcinoma, colon, glioblastoma, breast, lung, endometrial, renal cell, and head and neck. From these, we can begin to identify those targets, once again, that are differentially expressed in a tumor cell relative to its normal counterpart, as well as understand both therapeutic tractability, the expression of these protein targets in other normal human tissues, as well as to understand the potential use of these targets as part of clinical trial studies. This database enables the identification and validation of both new and established targets for cancer therapeutics, not only identifying the particular subsets of tumors in which we should enter into as part of a clinical study, but also allow for indication expansion across established targets. It allows to understand the distribution of a particular target within a particular tumor type, as well as the relationship between that target and particular mutational status. And this becomes very important in supporting patient selection and indication expansion once again. And finally, we can evaluate the expression of key targets in normal tissues to understand potential on and off target or on and off tissue effects that are central to the drug development program. So to summarize, proteomics has really undergone a transformation owing to a number of technical aspects over the last 12 to 24 months. This enabled the measurements of thousands upon thousands of proteins in a biological specimen at scale, finally enabling the study of thousands of samples at a given time. This, coupled together with the expansion in therapeutic tractability for particular agents, now going from several hundred proteins that may represent the druggable genome to over 20,000 proteins that represent the druggable genome, has transformed drug development as a whole. At Sapient, we can use our AI-based classification systems together with a generation of proteomics data to not only identify, but validate optimal targets. And in doing so, create enormous opportunity to discover new biological targets, to validate targets for existing therapeutics, and to expand indications for particular agents. I'm happy to go ahead and stop there, and time allotting, I'd be happy to take any questions. This is our contact information. I would welcome anyone in the audience to please reach out at any time if we can provide additional information. Our email address is discover@sapient.bio, and our website is www.sapient.bio. Our LinkedIn, our Twitter, and other social media information is also located here.
Thank you so much, Mo, for that wonderful presentation. I'd like to remind our audience that you can still submit questions by entering them into the Q&A box at the bottom of your screen. But in the meantime, we do have a couple of questions for you, Mo. The first one is, can you go back to the evolution of proteomics over the last several years? How has the field changed, and what is possible now that wasn't possible previously?
Absolutely, Stacey. I'd be happy to. Proteomics, particularly mass spectrometry-based proteomics, has always been the gold standard for measuring proteins, particularly not only measuring the native protein, but also the proteoforms that may be present in a disease state and the post-translational modifications that may occur in that protein. The challenge has really been one of depth and scale and robustness. In that while proteomics worked quite well, particularly again, mass spectrometry-based proteomics works quite well in a single sample, being able to expand that to thousands of samples and simultaneously measure thousands of proteins in each one of those samples in a robust nature over time and space, operators, instrumentation has always been a great challenge. Again, owing to some of the advances that have occurred in protein isolation using nanoparticles, that have occurred in the hardware around instrumentation for proteomics, and then in the software, I'm really excited to say that these limitations have largely been overcome. This enables us to measure thousands of proteins now across very large numbers of sample sets. And in doing so, we can actually use intelligent design to actually understand what represents an optimal target for a given therapeutic.
Great, thank you. Next question. How does proteomics intersect with genomics and transcriptomics, particularly within the context of multiomics?
Yeah, absolutely. And so I think there's been an evolution that's been occurring, Stacey, over the last several years of time where, whereas there was initial great dependency on a single data modality, whether it be genomics or transcriptomics or even proteomics, we've now understood that being able to integrate this information across these different data modalities is absolutely essential. Being able to understand the genome and integrate that with metabolite and lipids together with proteins and transcripts within the context of clinical outcomes or preclinical data is really what's required to be able to advance forward this field in a very meaningful way. So when we make these proteomic measurements, it's rarely in isolation. We often think about the proteomics and how this may map onto various transcriptomic signatures, how this maps onto genomic mutations, how this may intersect with particular small molecule mediators, whether they be metabolites or lipids, once again in the context of clinical outcomes data or preclinical data. And so there really is quite an opportunity here, as you can imagine, to be able to integrate this multidimensional data for diagnostics, for prognostics, for target ID, for preclinical development and drug screening, all the way up through companion diagnostics as part of clinical trials.
Great, thank you.
Of course.
Next question. How do you bring the quality component to this high dimensional data, especially if the data is used in later stages of drug development?
Yeah, that's a great question, Stacey. And really the goal here is not generation of large amounts of data for the sake of filling hard drives. And for this data to become valuable and useful for these approaches and workflows to really enable target identification and validation for which the entire subsequent drug development pipeline is dependent, this data has to be extremely robust and high quality. And as I mentioned earlier, there's been oftentimes, or with traditional mass spectrometry, critical drifts that have occurred in instrumentation and signal quality, in drifts in chromatography, et cetera, that have really limited discovery. And as we've developed these workflows, there's always been a key eye towards ensuring that the quality of the data that emerges is absolutely of the highest order. What we assay today and measure today has to be reproducible when we make that same measurement tomorrow or 1 year from now or even 10 years from now. And that's central to our process. As part of these development workflows, and I went through this very quickly, we've implemented very strict quality matrices that includes off-target samples, blinded replicate samples, spiked in standards into each biological sample. And this is what enables us to follow in a very robust manner quality over a given experiment, over a given sample, and over a given period of time across operators, instruments, space, and time. And this becomes really, really critical for ensuring the robustness of the underlying data that's generated. And with that high quality data comes the ability, once again, to leverage this information for knowledge generation and for target identification.
Great, thank you. Can you speak about the use of proteomics as part of clinical trials and its potential uses in the trial space?
Yeah, absolutely. So we briefly touched upon this, but when we think about mass spectrometry and some of these omic technologies, Traditionally, they have been limited to early target ID and validation, and certainly that was the context for the conversation today. This has evolved very importantly, though, in mass spectrometry and omics as a whole is no longer limited just to target ID screening and preclinical samples. This has very much evolved now into a place where this is central to clinical studies as a whole. This involves everything, once again, from stratification of patients, clinical trial design, understanding target engagement and pharmacodynamic markers, all the way through companion diagnostics. But it's becoming much, much more common as opposed to the exception, whereby omics technologies and mass spectrometry are being leveraged as part of those clinical trials. As Sapient, we have a CLIA CAP accredited laboratory that's central to this operation that allows us to take our discoveries and develop CLIA assays that can be leveraged as part of clinical trial implementation. And so this is a really key part of what we do. And that comes from being able to not only make discoveries, but rapidly translate them in a way in which they can be part of the clinical study infrastructure.
Great, thank you. How does proteomics inform on potential safety outcomes, particularly on tissue and off-tissue effects?
Yeah, absolutely, Stacy. So this has been an area that's been of great learning over the last several years as these new therapeutic modalities have really come into the limelight, particularly around radioligand pharmaceuticals, ADCs, T-cell engagers, et cetera, in which the specificity for a particular target is extremely high. The challenge is that these therapeutic modalities can oftentimes interact with that same protein target in an alternative tissue type, whether it be the heart or the liver, the kidney, the lung, the brain, et cetera. And as you can imagine, given the toxic nature of many of these therapeutics, particularly in the oncology space, this results in very profound on-target off-tissue effects, meaning it affects a tissue alternative to the tumor in a very negative way. This drives many of the adverse events that we've seen with many of these emerging therapeutics. So as part of target identification, we're very quickly and very careful to not only understand the differential expression of a particular target in a given tumor or in a given disease setting, but also to understand what the expression of that protein is across other tissues within the human body. This allows us to predict what may be some potential off-tissue effects. And that becomes really central for go, no-go around particular therapeutics, around particular targets, as well as for understanding and mitigating those risks going forward as part of drug development for our clients.
Great. I think we have time for one more.
Sure.
How is AI transforming proteomics?
Yeah, so there's lots of different ways in which AI and advanced computational analysis has really been central to proteomics. From a technical aspect, you can imagine machine learning and AI is central to just the analysis of proteomics data, being able to extract this information from a mass spectral file, being able to map peptides back to a protein and being able to quantitate these in a very accurate manner. Beyond just the technical aspects, as I mentioned, given the fact that we now can generate enormous amounts of data, whether it be in the preclinical system, whether it be in target identification, or whether it be in the clinical trial, being able to make sense of that tsunami of data becomes very, very critical. And this is where AI is really has enormous value. When we hear much about AI and drug development, oftentimes it's on the drug development side where we're using artificial intelligence to really inform on the best chemical agents to attack a target. Here we're using AI on the alt side of the coin in order to identify the ideal targets. In both scenarios, you can imagine it really is leveraging these ability of these advanced computational systems to take an enormous amount of data and make sense of that information in a way that data can be leveraged for knowledge and wisdom in order to drive forward a program.
Great. Well, thank you so much, Mo. It's always a pleasure to have you and Sapient partner with AAPS. This has been a wonderful presentation. And thank you so much. Any last words?
Thank you to the audience. Thank you to the AAPS and you, Stacy, and we really look forward to hearing.
Thank you so much. And if anyone wants to get in touch with Mo after this presentation, the emails are on the screen. Thank you again so much, and I look forward to seeing you all again soon.